University of Illinois.
Illinois. You're from the USA?
Yes.
Okay.
My accent tends to give
Yeah.
it away. And you're from Bogota?
No, I am from. I I work in Medellin, but I from Cartagena.
Ah, okay.
Do you know Cartagena?
I've heard of it.
Yeah,
But
of course.
yeah, I couldn't tell you the right number.
You have the presentation in what?
PowerPoint.
PowerPoint?
Do you know your list 20 minutes? The question is saying that maybe.
Okay. And what are the lights? How many lights do
Okay.
I have on?
Green is you have
Yeah.
10 minutes. Yellow is have 5 minutes for finish the presentation.
Ah, okay, so 5 minutes.
Red?
Red have been 20 minutes. Yep. Yep, it looks okay. Slightly cut off, but yeah.
Yeah, but for the
Yeah.
okay.
Great, thank you. Oh, can I get that back? Okay.
Lo máximo hasta poder hacer. Pero siguen las tres conjuntas y así así. 50 minutos. Pero igual que sí, o si quieres el inalámbrico.
Quizá para moverme.
Sí.
Bueno, buenas tardes a todos y todas. Ustedes saben, yo soy un alemán que vive desde hace años en América Latina. Por eso yo me presento a mí mismo. Próxima sesión voy a hablar un poco no sobre ciencia, sino sobre management of science cooperation in Latin America. So I will continue in English because what I'm presenting maybe could also of interest for all the colleagues coming from the different Max Planck Institutes. So, you know, my name is Andreas Trepte. My office is in Buenos Aires. Buenos Aires because there is the unique partner institute of the Max Planck Society. So if you are thinking about careers at Max Planck, you have learned there are a lot of institutes in Germany, several in other countries, but the nearest one first is Florida, is in the Americas. And the next one, not as an original Max Planck Institute, but it is the partner institute of the Max Planck Society for Biosciences in Buenos Aires. In the same building is my office. And well, I will start talking because I'm talking a little bit about also the history of my work, because we started in 2013 with Colombia, with a visit of the president of the Max Planck Society in Colombia, and as a result you will see from nearby nothing in the cooperation, we have established something, and I hope that we could continue growing in the relation with you. Again, as I said, the majority of the institutes, 79, are distributed in Germany, most of all nearby to the campus of a university. It's also important for cooperation, but also if you would like to make your doctoral degree, this is a doctoral degree you will receive in Heidelberg from the University of Heidelberg, in Berlin from one of the Berlin universities, with a stamp also from the Max Planck Institute, where you will do your research work. International cooperation, why? Because when I entered the Max Planck Society 28 years ago, the majority of the directors were Germans. The big majority of doctoral students were Germans. Today, starting from the top, 40% of directors are foreigners, or people with a passport from another country. Postdocs, the workhorses in the laboratories, group leaders, 85 to 90%. Doctoral students, 60%. So it's not necessary first to learn German to come to a Max Planck Institute. You need English, because your neighbor will be a Russian, or a Korean, or a scientist from India, and not a German. Or maybe the technician is a German, or the secretary of the director. But in general, you should know we are one of the most internationalized institutions. And this is at home, inside of the institutes, and this is abroad. Therefore, we learned with Latin America, normally the Max Planck Society, our institutes, we have really few agreements. We learned here, we heard today something about law, the rule of law in Latin America. For the universities, this means if we would like to cooperate as Max Planck with the university, first we need a framework agreement. We need 6 months or more to come to a framework agreement. Later we need a more detailed cooperation agreement. All this normally we don't need. But we have now several dozens of agreements, not only in Colombia, but all over Latin America. Of course, we have a lot of ad hoc, I name it ad hoc research projects between people, because one have the organism, the other have the analytics or whatever. They are publishing jointly, but there is no institutional link. It's only between friends, colleagues. So, we are today in the middle of something here, an exploratory symposium, because we are trying to look where people at Max Planck and in Colombia are working at the same time, more or less at the same topic, and could there be any kind of synergy? But internationalization of research means today there could be joint fellowship programs, for example with Ciencia in Colombia, or with the Future Ministry. There could be a call for projects, Colombia, Max Planck, so that starting cooperations could be deepened, could be developed. And, as you know, we have several joint, associated research groups between Colombia and Max Planck Institutes. But you see also, this requires a lot of coordination, and you know your country, you know your continent. Communication, regular visits are very important. So if you look in detail, and this is also the part, let's say, with information for young people which are really interested. All Max Planck institutes normally are making calls, individual calls from projects, because they are looking for PhD students. Organized calls from the so-called international Max Planck research schools that are graduated schools for doctoral students. All this is published in the internet. we are receiving all over the institutes a lot of applications, but only a few one from Latin America. So my comment is, please help me to raise this number. You will see, or you could see it here in this, oh, hold on. You could see it here. 130 Colombians at today, more or less, are at Max Planck Institutes. But it could be much more, so some are finding the way, some are trying to enter one of these research schools. And there are also now, they are starting now, just now, 3 new Max Planck schools. You could think in something like Harvard Business School, Harvard Law School, you have now also Max Planck schools in very interdisciplinary areas like cognition is one. The other one is matter and life. So at the interface between biosciences and natural sciences, exact sciences, or photonics. The same is true what I said. It is published in the internet. It is published on the web pages of these institutes when they are looking for postdocs. But again, these institutes are different from, let's say, a research center, un centro de investigación in Argentina, Chile, or in Colombia. Why? Because Max Planck Institutes are funded by the Max Planck Society. And they have an internal structure and a budget which allows them to realize their research program without third-party funding. Though they have their own stuff, they have their funds for cooperation, their funds for stipends, their funds to buy new equipment. So they are much more flexible than, for example, universities like yours. Because you have to wait, scientists have to wait for the next call from Ciencia or from the future ministry, or maybe a project in the European Community Program. This is not the case at the Max Planck Institutes. Of course, scientists at Max Planck Institutes could apply, and they are doing this very successfully, in external programs. But with these special conditions, they are not only very autonomous, they are very fast in a lot of tasks they are fulfilling in research. So if you would like to enter into an institute, there is this formal way. But for example, for postdocs, there is always the way to propose a project, to look in which institute this kind of research is done. Is my project bringing also an added value to this institute? And to send a proposal to the director or research group leader in this institute. You could do this always, in any moment. So, I mentioned there are spontaneous, organized research projects with Latin America, but I have to comment. Every year, Max Planck Institutes are maintaining 4,000 or 5,000 of these ad hoc research projects, and only 150 are with Latin America, and 10%, or less than 10%, 14 were with Colombia last year. So there's a lot of room where new cooperations, new links, new exchange of young people could be developed. But again, we are here to inform you, the speakers, the representatives of universities, but now la pelota está en el campo de Colombia. You have to act. You have to take the initiative. You have to look into the internet. When will the next program in solar physics start in Göttingen? And then prepare an application. Nobody after this symposium will contact you again. Yeah? So please take this into consideration. So, plan for next year is really to have a joint call with Ciencias, and I hope that the new ministry will not only be a new institution, but also will have a new, bigger budget, than Ciencia today. We have two different kinds of research groups. For those which are doing their postdoc, because a person with a doctoral degree is not qualified, let's say, to be head of a research group. We need more experienced researchers. And there are two categories. One is the so-called partner groups. This is a program of the Max Planck Society for those postdocs, for example, the Colombia, which would like to go back to their country and which are really excellent in their scientific output during their research time at the Max Planck Institute. Minimum 1 year you have to be at the Max Planck Institute. Then, if your director would support you, you could apply during the year in this program. And you will see, I have mentioned it. The first group has been established at the University Uni Norte in Barranquilla. So, si se puede, se podría decir, no? We could say, it's possible. But it is a solution for persons. And this person, as a postdoc, is a specialist. So he is also defining the topic, and he's defining which institute could be, or which university, which faculty could be then the host for this group. But we have a second category, which are the tandem groups. And with these tandem groups, we started in Colombia because there were joint thematic interests, but no postdocs from Colombia. So one way is we know the person, and we help the person that this person could reintegrate in his university or in a different university. The other way is there is a research topic where one or several Max Planck institutes and a university or research center in Colombia or in other countries would like to cooperate. Then, in the case of Colombia, we are organizing an international call. We are selecting between the applicants, the best candidates, and in a selection symposium, in live interviews and presentations, the candidates, or let's say the final candidate, the future group leader, is selected. So I hope you understand, we are an institution which is not a university. We are very decentralized, but there are a lot of opportunities for doctoral students, for postdocs, and even there is a support to come back to the home country, in your case to Colombia. So here see the names of the and topics of the 6 groups which are at the moment working at the National University, at the University of Antioquia, Los Andes, and Cali. We hope that the number of groups will increase, but this depends from the research interests of our institutes and also from the initiative and the availability of funding in Colombia. So, during the last 6 years, you see, we infected a little bit Latin America, because what you are seeing is the development of research groups headed by former postdocs or internationally recruited postdocs as leaders for tandem groups and research groups, partner groups in Latin America. So for me, for next year, we would like to negotiate with the ministry in Colombia a program for fellowships, because the institutes, Max Planck Institute, are open. And as I said, every year so many programs for doctoral students are open. There are fellowships from the Max Planck Society, but the majority is financed by fellowships from national institutions like Ciencia or German institutions like the German Academic Exchange Service, DRD, or the Humboldt Foundation for Postdocs. Well, we are inside of the symposium, and you see we are trying to extend the network of corporations. Next candidate could be the Universidad del Rosario. So, to resume, all the information what I am presenting is normally available. And as you know, I am living in between Latin Americans, so I know English is a challenge. It's a challenge to take the initiative, but I could only say, we offer you at this Spanish website, which is handled by my office, all informations about upcoming calls in Max Planck Institutes, which could be of interest for you. For you means for young people, excellent master students, which would like to make their PhD in one of these programs of Max Planck, or for established profs, young profs, which would like to make a postdoc in a Max Planck Institute. So this page is available. We have the same information for those which are preferring this kind of US-American communication in Facebook. So you find us also there, and you know our office, we are in Buenos Aires, we are speaking German, English and Spanish. So please feel invited to contact us so that we jointly could develop the cooperation, but always you should have in mind, Max Planck Institutes are leaders in their research field at the international level. A cooperation with Colombia or with whatever country on the world, whatever institution, should also have a value added for our institute. So the proposals should be really at the frontiers of science, but the institutes are big. We are in total some 23,000 people. So feel invited to be part of this endeavor at Max Planck. Thank you so much, and here's my contact email, and feel free to ask questions if you like. Thank you so much. If not, we continue with the scientific talks. And again, thank you.
Hi, my name is Isaías Lance. I am the chairman of the next session. This is the Microbiome Science session. For this session, we have two speakers. The first one is Gillian Waters. Gillian gets his PhD from the University of Illinois, and now she is part of the Department of Microbiome Science of the Max Planck Institute for Development Biology. Now, Gillian will present us the interplay between host genotype and gut microbiomes, impact on diet and health. Gillian?
Thank you. And here's this. Hi, thank you. It's great to be here, and I'm excited to tell you a bit about the work we do in our department. So first of all, here is a nice view from our rooftop terrace. So Tübingen is very beautiful and hilly, and fortunately our institute is at the top of one of these hills. So we have a great view there. So our group is relatively new at the Max Planck Institute. So we just moved in 2016, and before that we were at Cornell University in upstate New York, where Ruth had first started her own research group there in 2008. So Tübingen, if you're not familiar with this, is, I don't know if that's very accurate, but it's sort of the southwest corner of Germany. We're a little bit south from Stuttgart, and our Max Planck campus has a total of 3 institutes. So I'm at the Institute for Developmental Biology, but we also have cybernetics and intelligent systems. So technically there is the Friedrich Mischer Laboratory, but in reality this is just a part of the Institute for Developmental Biology. So our institute has a total of 6 directors. Some of you may think I can't count because there are 7 faces up here. Felt like it would be a disservice not to put Christiana Nusslein-Fullhard up here. So she retired a few years ago, which is when Ruth took that director position, but she does still have an active research group at the campus. One of the things, though, is that right now we're really in a phase with a lot of turnover. So in principle, we really have 4 directors at the time, currently, excuse me. Unfortunately, Elisa Izaralda passed away unexpectedly last year, and Gerd Jurgens is retiring, so we're actively searching for 2 new directors. And the Institute is sort of changing a research focus, so we're really moving more towards host microbe interactions, as that is a common theme of some of these other research groups, and given our name of microbiome science, it's probably not surprising that we're interested in that as well. At the moment, we have a total of 7 independent group leaders. These top 4 are part of the FML, and we currently have 3. So again, a lot of turnover even with the group leaders. So we had 4 that just ended their stay last year and the year before, so I anticipate that we'll probably have a few more joining us soon as well. So a little bit about us, Department of Microbiome Science. So first of all, in case you are not familiar with the microbiome, to make this very simple, the microbiome are the microbes that live on us, so that could sort of be on the skin, and within us, so my favorite part is within the gut, which I'll tell you more about. So the microbes, in the microbiome field, we have a tendency to focus more on bacteria and archaea for the ease of cataloging these microbes, but this would also include eukaryotic organisms, viruses as well. So there's a couple things that I would want you to take away about the microbiome. One is that we really are heavily dominated by microbes. So the estimates vary, but for every cell on our body that is ours, that is a eukaryote, there's at least 1, if not up to 10 times as many microbes, suggesting that we're just as much microbe, if not more microbe than human. But what's important about that is also the gene content that they provide for us, because this really influences our health, in many positive ways. So the genetic content is about 10 to 100 times the amount of what our own genome encodes. And one of the principal functions that comes to mind, for example, is the ability to help us digest a lot of the complex polysaccharides in the diet that we don't have the enzymatic capabilities to do so with our own genome. So although today I'll be telling you more about host genetics and the microbiome, we really do have a wide array of research projects within the lab. So a few examples are the gut virome. We also look at bacterially produced sphingolipids and how this influences skin development, brain development. We don't just do humans and gut, we have maize and Pseudomonas. We have this really neat organism that seems to be able to live in seawater as well as the gut of polar bears. Diet and evolution, we're also very interested in methanogenic archaea. But unfortunately, I don't have the time to tell you about all of these things. So I'll give you a couple examples of what we do in the lab. So one of the things is we know that the gut microbiome does directly impact host phenotype in a number of ways. So the example I'm showing you here is with obesity, which is one of the most robust associations with the microbiome in human health. I would say inflammatory bowel disease is arguably about at the same level of importance. So the gut microbiome can influence host metabolism, so this can contribute to insulin resistance, metabolic syndrome, and this is caused by the microbiome through inflammation and basically the metabolites that can be produced by these microbes in the gut. So conditions such as obesity also have a very strong genetic component to them as well. And another thing a question that we had in the lab was sort of what influences the microbiome, and so who we have in our gut, and basically the relative abundance or how many of them are relatively there. So one of the questions that we had a few years ago was, well, how does the human genotype affect the microbiome, which we know impacts host phenotype? So this was a study of heritability, which is not to be confused with inheritance, which can be vertical transmission, but this is more a genetic predisposition. So these are usually done with large twin-based cohorts, and so we did this with a number of twins from the UK. cohort name is Twins UK, not too unique there. But so one question, so oftentimes people will say height runs in families. So often, if you were to take a twin, if one twin is tall, the other one is likely to be pretty tall as well, because there's a strong genetic component to that. But one question was, so we could look at the microbiome as a quantitative trait, and ask that same question as well. And so not only the microbiome in terms of composition, but also the components or sort of function that are coming with this as well. So heritability is the proportion of variance in a quantitative trait that is attributed to host genetics. So unfortunately, I'm glossing over a lot of results here, but one of the things, so I said we had the question, how is the microbiome influenced, and what members of the microbiome are more or less influenced by host genetics? And this family Christensenalaceae was the most highly heritable, meaning that about 40% of the relative abundance of this bacterial family in the human gut can be attributed to host genetics. And one of the things that we also had some information, so we had BMI, or body mass index, information on these twins, and we saw that lean, healthy BMI individuals had more Christensenalaceae compared to obese. So that, and again, with some other results that I don't have the time to tell you about today, we wanted to ask the question, is this just an association, or could there be a direct causal effect? So one of the ways that we can study the direct effect of microbes on the host is through fecal microbiome transplants. So maybe you've seen this in the news, for example. So this is done with humans. Oftentimes this is done for the treatment of Clostridium difficile infections, which are usually a consequence of inflammatory bowel disease. This isn't just any sort of normal diarrhea. It is really horrible. It is extremely difficult to treat and may not always respond to antibiotics. And often antibiotics are what caused this in the first place. So basically there's multiple ways one could do the fecal microbiome transplant in humans, but what's pretty great is there is a very high success rate with this. And we can also leverage this same method in a mouse model to study the microbiome as a direct effect. So as I said, one of the questions I had was I wanted to see, well, does this Christensenalaceae family promote leanness? And so one of the things is that with obesity and the microbiome is we know, so I told you before that the gut microbes do directly influence phenotype, especially with respect to obesity. So we know this, not just us, but other research groups have done this as well, is that if you take a germ-free mouse, so these are mice that are 100% sterile, they're raised in bubbles, and if we take these same mice, they're reared in the same lab environment, they're eating the same diet, so it's a very well-controlled environment. And if we take stool from an obese human donor, it sounds gross, but mice love eating feces on their own anyway, so they do this gladly on their own accord. These recipient mice will have an increase in body fat accumulation, and sometimes we can also transfer impaired glucose metabolism and dyslipidemia to these mice as well, simply through the microbes from an obese donor. Conversely, mice receiving stool from lean hosts often stay nice and lean and a normal weight for a mouse at that given age and strain. So the question I had, so with this family Christensenalaceae, so I did an experiment where, so having obese donors that had very low levels of this family, so I predicted that obese donor stool would result in a heavier mouse. So I wanted to ask the question, what happens if I add in Christensenalaceae? So at the time, I was very lucky. There happened to be one cultured representative from this family. So I ordered this from the DSMZ, added this to this exact same obese donor stool to ask the question, if I add in Christensenella minuta, will I receive a leaner mouse? The answer to that was yes. So I was very careful to treat this entire community so they were getting the exact same treatment, except with the exception of having added Christensenella minuta. And so this is 3 weeks after inoculation. As you can see, the mice receiving Christensenella gained less weight, and the body fat percentage was also less in this case. So this is what I've been working on in lab, and this is my very large research group at the moment. So since I'm relatively new at the Institute right now, it is just myself, and I have one PhD student. And so the focus of my work is really following up on this. So the questions I have are, why are these mice putting on less body fat with this treatment? So what's going on in the host? how does this addition of Christensenella minuta affect the rest of the ecology of the gut microbiota in these mice? And then what does Christensenella do? Because there's very little out there that's known about this bacterium, and it's because it's a relatively new family. So as much as I would love to tell you all about my research, I figured I should give a broader representation of the department. So also on the theme of host genetics, so one of the questions, so I told you that Christensenalaceae is a very highly heritable family, but what gene or genes are driving this association? And the exciting answer to that is, I don't know. So we can assess, so we can do genome-wide association studies where we look at single nucleotide polymorphisms and associate them with health, or in this case, we wanted to look at this with the microbiome as a trait. So if any of you have ever sent off a saliva sample to 23andMe, this is basically the same information that they're taking to tell you if you have a genetic predisposition to a certain disease state. So unfortunately, just because we have to make so many statistical comparisons when there's so many SNPs, that we just don't have the sample size to have the power to detect which gene or genes are influencing Christensenalaceae. But one thing that has come out of these genome-wide association studies, not just from our group, but from others as well, has been an association with lactase persistence and bifidobacterium. So lactase persistence is associated with lactose tolerance, and this has a genetic component and it varies geographically. So some populations have very high levels of lactase persistence. So this is basically, lactase is the enzyme that digests lactose. And so all of us, being mammals, have the ability to digest lactose at a very early age, but for some people, the ability to do that is lost over time. So basically what happens if you persist, the lactose, is broken down by lactase, and the host, if you're a lactose tolerant person, gets a bit of energy from that lactose. Conversely, if you're lactose intolerant, you don't have the enzyme, or making this a little simple, but basically the lactose can go through the gut, it's utilized by microbes in the gut, and this causes gas production, which is then what causes a lot of the symptoms of lactose intolerance. So if you're someone that is lactose intolerant and you're taking Lactaid, this is really just the enzyme lactase. So bifidobacterium, which this name may sound familiar to you if you're not a microbiologist, so this is something that is often used in probiotics. It's a type of bacteria that is used in a lot of yogurts, and this is increased with lactose intolerance. So as I just said, what we suspect is that because if you are someone that doesn't have the lactase yourself, it's going through the gut, bifidobacterium loves it, and then again, this is what can result in these symptoms. So one of the things is that, so I'll tell you about a study that we're doing in the lab, and of course just asking someone, how do you feel after you've had lactose, is not the best question because that's very subjective. But we can analyze this quantitatively by looking at two things. So the first is the blood glucose response. So this is sort of assaying the host response to lactose. I should also say too that in this case when we're assaying this, a person is fasting overnight, about 12 hours, and then on an empty stomach, just eating pure lactose, because stomach emptying and things like that can also influence the glucose response. So as I just said, so if you don't have the lactase enzyme, you won't have a blood glucose spike because you're not digesting it yourself. It goes through in the gut, and sorry you probably can't see this picture. In case anyone is wondering how are we measuring gas output, this is through the breath. And so we're measuring hydrogen and methane production. And conversely, someone that is lactose tolerant, they do have a spike. Sometimes it's mild to modest, depending on the functional capacity of their lactase. And then usually they have no gas production. So one of the things is that, there's a few people in our lab working on this project, and this was in collaboration with a lot of other places. So this is before I showed you the cute, sad cheese map of lactose tolerance. So here's a more quantitative one. And so basically this is a proportion of lactase persistence within a population. So we had 3 study sites. So the first here was in Germany, where lactase persistence is pretty high. Most people are tolerant. There was a study site in Vietnam where it is relatively low, and then in Gabon, which had an intermediate effect. And so at the site, Victor and Liam, as well as with local researchers at these study sites, they had about 200 mother-infant pairs. They did a wide array of characterization. So they also did the genetic testing to see what their genotype would be, these blood glucose and gas measurements, blood measurements, and many other things as well. So one of the things that's been really interesting, and not surprisingly, so we had some people that are lactose tolerant and some that are not. But there was a third phenotype that came out of this, and this is what they call acquired tolerance. So in this case, so these data are still coming in, this is relatively new, but one of the things is, so basically what I mean to say is I don't know the genotype of these people that I'm saying have an acquired lactose tolerance because that data is not in yet. But when it came to a blood glucose response, there was nothing, no spike. So as I just told you before, this would suggest that they are lactose intolerant, but there was no gas production either. So the big question from this is, oh, sorry, I thought I had another thing there. Where did the lactose go? And again, I don't know the answer to that at the moment. As I said, this is ongoing research, but I wanted to show you some newer unpublished results as well. So figuring this out is definitely something that many people in the lab are working on. So one of the things will be, for example, to feed C13 lactose. Ideally, this would have been nice to do in humans, but it's rather expensive to do the heavy isotope labeling with the lactose because of the quantities needed, because when humans are doing this, you're taking about 25 grams. So instead, this will be done in bioreactors. And because of the effort that it took to get these samples and these questionnaires, so many other projects will come out of this cohort as well. I realize I went through this kind of quick, but that is a good thing, so maybe we can kind of catch up a little bit. So I just want to acknowledge I work with a fantastic group of people, Director Ruth Lay, Alejandro Reyes, who is the Tandem Group leader as well, and of course thank you to all of you for staying here and for your attention today. Thank you.
Thanks, Gillian, by your nice talk. Now we can move on to the next speaker. The next speaker is Fabio González. Fabio is part of the Instituto de Ciencias Naturales de la Universidad Nacional de Colombia. Fabio will present us the developmental biology of non-model tropical plants, case study in early floral evolution, convergent fruit evolution, and plant parasitism. Fabio?
Thank you. How will use this one? Good afternoon, everybody. Thank you. No? Yeah. Okay. Gracias. Many thanks to the organizers for the invitation to participate in this symposium. It is really my pleasure to tell you a little bit about the research that we do in our Plant Evo research group. This group was established like 5 years ago as a collaboration between the National University of Colombia here in Bogota and the Universidad de Antioquia in Medellin. Our main research topics are focused on the study of the genetic basis of several developmental processes in tropical non-model plants, mainly related to flower and fruit evolution. Although species number is lower in early divergent lineages, we have focused on them because their morphological variations are in fact higher and somehow less understood, given that the majority of model plants are less variable, more predictable, and better understood. As I will tell you in this presentation, we have integrated several techniques, including virus-induced gene silencing, in situ hybridization, protein-protein interactions, and a large doses of transcriptomics. Let's start the talk by saying that the flowering plants have undergone at least 7 whole genome duplication events, here marked with the yellow stars, that have been critical for their evolution and diversification. For instance, our breakfast today is nothing else than a bouquet of polyploids. However, as I mentioned before, we are primarily interested in early divergent lineages that are not represented by the model genomes or systems for functional analysis, and those that predate the major whole genome duplication events in flowering plants. Our research group is interested in addressing general questions in evolutionary developmental biology. For instance, one of these questions más bajo, más despacio. Primera vez que me pasa en inglés, perfecto, mucho mejor. ¿Me devuelvo o no? Uy. Okay, let's start here again by saying that flowering plants have undergone at least 7 whole genome duplication events. Here, you can see them with the yellow stars in the cladogram, which have been critical for their evolution and diversification. For instance, our breakfast, I told you, is a large collection of polyploid plants. However, our main interest is in groups that predate those whole duplication events in the genome. For instance, one of the questions is why plants such as the early divergent genus Aristolochia, which you see here, have taken on such amazing floral morphology variation, perhaps more so than any other group of flowering plants. Aristolochia comprises about 600 species with an amazing floral variation, not only in terms of floral shape, size, color, but also in terms of epidermal elaborations that are unique in the plant kingdom. Thus, we have focused. Can we play the video here? On the so-called matchbox genes, those that are genes responsible for floral organ identity as a proxy to understand the evolution of these flowers. Aristolochia perianth is differentiated into 3 portions, an inflated one at the base called the utricle, followed by a narrow portion named the tube, and an apical portion which is called the limb. This last one expands at maturity basically to seduce pollinators. Colombia has the smallest and the largest Aristolochia flowers worldwide, varying from about 1 centimeter in the tiny Aristolochia numularifolia in the Orinoco Plains, by the way, collected for the first time by Humboldt and Bonpland, to flowers of about 2 meters long in Aristolochia grandiflora, which inhabits Chocó and neighboring areas. Despite this extreme size variation, the cell division, expansion, and differential growth are genetically regulated by the same genes, some of which have actually not been reported in driving floral symmetry or perianth identity elsewhere. We have applied in situ hybridization to closely examine such genes. This is a technique that allows to detect where and when a gene is turned on, the purple areas in the slides. Here I am showing you the expression of matchbox genes responsible for floral organ identity in Aristolochia fimbriata. Whereas the B and C class genes important for stamen and carpel identity in Aristolochia fit the model predicted from Arabidopsis, we found little conservation on the genes that are important for periant identity, as they have broad expression patterns very different to what the model predicts. The good news is that we found an alternative, non-canonical gene that appears to be responsible for the identity of these bizarre flowers. This is in fact the gene agamus-like 6, also known as AGL6, which so far has never been associated with periant identity. In addition, by implementing a protein-protein interaction method using yeast-to hybrid, we were able to identify that the formation of an AGL6 sepalata tetramer is in fact required to establish periant identity in Aristolochia. The second question we are addressing at the moment is the control of the bilateral symmetry in flowers. This trait is critical to successful pollination. Now we know that bilateral symmetry in Aristolochia flowers follow a different gene mechanism to that known to occur in, for example, orchids or snapdragons, which have been the model to study bilateral symmetry. In green, I am showing you the TCP transcription factors that are important for reducing cell proliferation on one side of the flower with respect to the other. We, in fact, have been able to find that genes known as Cincinnata genes, which are also from the TCP family but are not the canonical cycloidea genes from the model species, are responsible for such bilateral symmetry in Aristolochia flowers. And again, this is a function that has not been reported before. Our question number 3 has to do with the genetic mechanisms shaping the different portions of the flower. We have also studied differential gene expression in the 3 floral portions, whereas the limb and the tube have specialized hairs to trap the insects. The utricle is covered by nectar-secreting hairs to feed the trapped pollinators. For this, we generated individual transcriptomes from each portion of the perianth. Through this approach, we were able to identify the top 20 genes differentially expressed between the limb, the tube, and the utricle. Among them, Cincinnata genes also appear to play a role in cell proliferation. We have actually been able to find also a microRNA 319, which is a post-transcriptional regulator of the gene Cincinnata 1, and we have validated this finding via real-time PCR. Now, let's move to the fruit projects we have. Fruit development and evolution is as diverse and complex as flowers. We aim to study the genetic mechanisms that drive fruit development and evolution in 3 main groups of flowering plants: the poppy family, the tomato family, and the coffee family, 3 of the most important groups in terms of economic botany. In these 3 unrelated families, fleshy fruits evolved from dry fruits, and what we are asking is whether there are similar patterns or similar genetic mechanisms regulating these shifts. We hope to have some answers for our next binational meeting, hopefully. So far, we can anticipate that there are some genes required in all 3 case studies, but are others that appear to be species-specific. I'm going to show you one example in the tomato family. Here we have applied a technique called virus-induced gene silencing. This is a powerful technique that allows direct functional analysis. This technique takes advantage of the plant defense response to viruses. The target gene is inserted into a viral vector, That in turn is inserted into agrobacterium, which is used to infiltrate the plant. The plant actually responds by silencing the inserted gene, thinking that she recognizes the gene as part of the virus, not a gene. So the transformed plant shows a mutant phenotype, which is the result of the down-regulation of the target gene. Given that this transformation is transient, this is not fixed in the DNA at all. This is at the RNA level. The phenotype is not to be considered a genetically modified organism. By using this, we have been able to identify transcription factors that are important to stop lignification in both dry and fleshy fruits. When we silence these genes, we in fact get more lignification in the fruits. Finally, I will finish with the plant parasitism. Our interest in this topic is to examine the evolution of the plastids. That is, bacteria evolutionarily trapped since a long time ago in the plant cytoplasm. Thus, they are the microbiomes of the plant cells, which are, by the way, responsible, among other things, for the production of oxygen on Earth. We are studying plastome evolution at 3 levels of parasitism, from facultative parasites to my right, to holoparasites to the left. So far, we have sequenced and assembled the plastome of Castilleja paramensis, this tiny, tiny plant from the paramos, and the holoparasitic Pilostyles boyacensis, which grows around Bogota in very dry areas of the Cordillera Oriental of Colombia. The genus Castilleja was already included by Humboldt as a paramo element in his famous Geography of Plant chart. Our Castilleja paramensis can grow free or associated underground to another paramo endemic, Plantago rigida. Sorry. They can communicate by little haustoria between them and grow together, but Castilleja paramensis can also grow freely. Surprisingly, the plastome, which is the chromosome of the chloroplast, is actually larger in this plant than that of the, for example, Arabidopsis or other non-parasitic plants. That is an open question. Why is larger than non-parasitic plants? We still don't know the answer. On the contrary, the plastome of the holoparasite Pilostyles boyacensis, as expected, is very much reduced. Not only 7 genes, to only 7 genes, and over 12,000 base pairs, being one of the most reduced plastomes across the Planck kingdom. This makes sense, as our paramo ecosystems are quite young, whereas similar extreme plastome reduction in Pilostyles has been reported from African and Australian species, from which we can assume that this plant parasitism is actually one of the oldest on Earth. With this, I finish my presentation. These are the founding sources we have had in the 5 years of existence. And this is the happy family that have helped a lot in the projects we have at the moment. People from all over the world, New York, Valencia in Spain, Germany, and the rest are more or less Colombians. I just want to finish with this. This is Dr. Natalia Pavon Mora, who is the associate professor at the University of Antioquia. She is the co-leader of the group, and she says hello from Dresden, where she is now a visiting professor at the moment. Thank you very much.
Thanks to our speaker for the fantastic talks. Now we have time for several questions.
Okay, my question is for Gillian. First of all, thank you for your talk. It was really interesting. The first one is, you have said that inflammation is really important in this interplay between the microbe and the host and the increase or the state of the disease. But I'm sure that's not the same in any state throughout the life. So,
I would agree with that. I don't think inflammation
yes.
is everything.
Yes,
but that's one means that microbes can contribute.
Yes.
to
So my
obesity.
question is, have you seen these changes between the population among different age groups in order to see this kind of inflammaging state, like this concept of the inflammation associated with the different state in the aging process, you know?
So in our group, we haven't done anything like that. So when it comes to inflammation, we have done a bit of work looking at TLR5, which is a part of the immune system that senses bacterial flagellin, and looking how that contributes to host inflammation. But as far as looking, that's an excellent point, seeing how age and over time and whether that changes, we haven't done anything like that. So.
Okay, and the other question was, I know you said that you don't know what happens with the lactose in the third group of induced tolerant to that lactose, but I would like to know what's your hypothesis. I mean, what do you think that it's happening with the lactose?
That's a good question. So honestly, I have to say I probably haven't thought as much about that since that's not my project, which is really a bad answer. So my guess is that some other microbe is using it, and for whatever reason, this would be better if I knew much more about metabolic pathways in bacteria, but unfortunately, I can't name any off the top of my head that would say, aha, this is a possibility that makes sense. So this is exactly why I want to take a labeled lactose molecule and study this in bioreactors and then really see. So we could take human feces, put this in bioreactors, feed them with this labeled lactose and see so what microbes are incorporating that lactose. We could also do metabolomics and see what's produced. And so that, I think, is going to shed some light on that. But I'm sorry that I can't give you a better hypothesis at the moment.
No, it's okay. Thank you.
More question?
I have a question. You showed that with this transplantation of, I don't forget, with C, this...
Christiansenella. It's a mouthful, yes.
So one question is, I mean, in principle, this would mean everybody who's obese just gets this in a yogurt or so and then becomes lean. So how stable is this transfer, or is there then a mechanism by which these bacteria are then kind of killed and one goes back to the original situation? I mean, there must be some selection why certain people have certain microbes and why others don't, and what is that mechanism? I mean, it must be immunity, I guess, or some whatever these antimicrobial peptides or so that I think are created in the gut as well. But I mean, what's the host doing? I mean, apparently it's not just that. It's not just the kind of the interplay of or the composition of the microbiome, but the host must also have some role, otherwise probably.
Yes, we think so, especially since, you know, the work, and I should have also mentioned too, so this effect of heritability, so having a large host genetic effect, this has been seen in other groups as well using other cohorts, so it's not just something that I'm telling you that our group has found, but others have as well. So to get at the issue of abundance and whether it's there or not, so one thing is that from what we've seen is it looks like most people have Christiansenella or the Christiansenellaceae family of bacteria in the gut. And so in our Twins UK cohort, what we saw was about 97% of individuals harbored this bacterium. But my guess is that, so one thing is that this family is at a very low abundance in the gut. So we're talking 0.1, 0.01%. So those other 3% of people that we didn't detect it, it was probably just that the sequencing depth wasn't sufficient. So my guess is really that most people have it. To get at your question about what happened in the mice with this transplant, so in the inoculum, I added what I estimated to be about 10 to the 8th colony forming units. And so it's a very large bolus of Christiansenella, and I made sure that they had the same equivalent amount of fecal microbes in this as well. And what I quickly saw is that, so even 24 hours later, the abundance drops down much more, and by 3-5 days post-transplantation, I see what I would call physiologically relevant abundances of Christiansenella in the recipient mice. So they are persisting, but they're getting to these low levels that would typically be found. And I should say Christiansenella is naturally a part of the mouse microbiome as well, and many other. And I'm sorry, I think you had one other point in there that I have forgotten now that I didn't address.
Well, how is the regulation between the host and the...
Yeah, so the host and Christiansenella. So at this point, I don't know for sure what it is. So one of the things is, of course, as you can imagine, I'm very interested in this family, so I've been following the literature, and anytime there's citations with this family. So we do know, or there is a lot of evidence from other studies looking that it is responsive to diet as well, and that is one thing. So when I talk about these host genetics, we don't know what gene or genes are contributing to this, but there could be things, for example, genes that drive dietary preferences, and so those genes that change the dietary preferences could be what is indirectly or what is directly affecting the abundance of Christiansenella. So as far as inflammation, so I didn't talk about this, but of course in my endeavor to try and understand what's going on in the host, I did look for evidence of inflammation. So because one of the things was, so I first looked at chow consumption, how they're metabolizing the diet, short-chain fatty acids, and then one question was, well, what if instead of having this wonderful weight-reducing bacterium, I'm just making the mice sick? So to basically gloss over a lot of results in that, at least in this model, I don't think that's the case. When it comes to human studies, Christiansenellaceae is almost always associated with a healthy host. So this is especially relevant for like inflammatory bowel disease. It's basically always enriched. There's an increased relative abundance in healthy controls. Crohn's disease, ulcerative colitis, it's virtually undetectable. So I'm inclined to say, I think all the evidence that we have to date so far suggests that there's no inflammatory response to Christiansenella minuta.
More questions? No more questions? Okay, I would like to say thanks again to our speaker. Applause for. So now we have 15 minutes for a break. 15.
Hello, everybody. My name is Joao Muñoz. I'm here to tell you that now it's time to talk about archaeogenetics. And we will enjoy two talks. The first one is by Dr. Cosimo Post. He is from the Department of Archaeogenetics at the Max Planck Institute for the Science of Human History. And he's sharing with us a talk on the early differentiation of Central and South Americans. Dr. Post, welcome.
Good evening, everyone. Okay. It's great to be here, and today I'm going to talk a little bit about the Americas and the settlement of the Americas. And as every other person coming from the Max Planck, I have to start with the typical slides about our institute, how fantastic our institute is. It's in Jena. It has been already introduced to where Jena is, kind of middle Germany, in the center of Germany. And our institute is actually quite recent. It was established in 2014, and the work there began only around in the mid-2015. So it's quite recent, and as you can see, it is composed by two main parts. There is an old villa where the administration is, and also the IT department, and there is this new kind of building with the guest apartments up there. So with a bridge in between, kind of bridging, in fact, the old and the new together. And that's actually what we are trying to do in this institute, the Science of Human History. So a little bit about what is our research profile. We have three directors. One is Russell Gray, that is leading the Department of Linguistic and Cultural Evolution, to which Mattis List is part of, and is going to introduce the department later. Then there is Johannes Krause, that is the Director of Archaeogenetics, which I'm part of. And then we have Nicole Boivin, that she is leading the Archaeology department. And a little bit about the research profile. What is the main idea is actually to bridge the gaps between humanities and natural sciences. And to do that, we at least try to do some interdisciplinary research of human history, of course develop new methods, and in general study and research on different cultural as well as biological phenomena, and bring this information together to draw some inferences about our past. Okay, now I'm going to introduce what we are doing in the Department of Archaeogenetics, and we have two main lines of research there. The first one that you see there is the studying the genome of historical pathogens. What we usually do, we at least how we started, go into mass graves, mass burials where you have many skeletons buried all together, and extract the DNA from, for example, the teeth of these people, and try to see if we find, beside the human DNA, also the DNA from the causative agent of this of this pathogen, of this death. So why these people die? Which was the bacteria or the virus that killed this person? And then once if you are lucky and we find that, and we try to reconstruct the genome of these pathogens and also reconstruct their evolutionary history. And then, of course, we are really working in the direction of host and pathogen interaction, and see how, both the pathogens and humans co-evolved throughout time. The second really strong focus on our research, I would say the strongest because I'm part of it, but it's equally shared, is the human population genetics. And here what we are doing is extracting DNA from ancient human remains to get and to collect insights into the past population migration as well as admixtures. And we try to link in this kind of multidisciplinary framework the genetic information with archaeological or historical information, and in some cases also with linguistic information. And the main goal is, of course, to try to reconstruct the human population history through time. So just to show you a little bit about which are our facility is kind of CSI kind of work we are doing there. At the core of our facilities is so-called clean rooms. They are, and we are really lucky to have available this really large laboratory of 250 square meters. It's the largest ancient DNA laboratory that exists so far, and it's really good actually because. Okay, it's really a pain because you need to, of course, keep it clean, and the bigger it is, the more cleaning is necessary. But it's really good because this allows us to split our work in different rooms, and in each room we do a different step of the workflow. And in this way, we try to reduce cross-contamination between samples and also to contaminate with our own DNA the ancient remains that we're working on. And of course, then there is a larger part of our laboratory that is a molecular biology lab where we do all the amplification of the DNA, and then eventually we sequence the DNA on these Illumina sequencer that are really powerful machines that produce million or billion of DNA sequences in few hours. And here, just wanted to show you some pictures, some snapshots of how we work in the lab. These are pictures taken by a journalist from the New York Times. It was completely wrapped up to not only him, but also the camera to enter in the clean room. And that's kind of some ideas. We try to protect not ourselves, but actually the sample from contamination. We wear these suit, glass, goggles, masks, and the only thing that is not real about this picture is usually we actually keep the light on. It's not we work in the dark, but I guess it was scenic for the UV lights, the contamination. Okay, so what I want to talk today about is this paper that we published last year about reconstructing the deep population history of Central and South America, really bold title. And of course, it's just the beginning of this research. And one of the reasons why I'm actually really happy to be here is that we are really trying to expand further this research in different regions and different temporal horizon. And the settlement of the America is a really long debated question, and so far what we know from the genetics, both modern and ancient genomes from Native Americans, is that the source population of non-Arctic populations, so the population that are excluding the Arctic one that receive later population movement from the Inuit, for example, the Eskimos. But the non-Arctic population of the America derive from one major gene flow event from a population that is formed by two distinct genetic ancestries. One tracing back to an Asian population related, for example, to Han Chinese, and another one, a population that was discovered in Siberia on the Baikal region and has as a proxy an individual hunter-gatherer individual from 24,000 years ago that represent a good proxy for this, let's say, Siberian population that mixed in the past, more than 20,000 years ago, with this East Asian group to form the source of this major migration into the Americas. And as you can see here, once the population, this stream, this group of people managed to actually enter in the Americas, they rapidly split into major groups. One that is here giving rise to the genetic ancestry present in northern Native Americans and Athabascans, and the other one that went down and provided the genetic composition of Central and South Native Americans. The same year of this paper was published, there was another paper that I want to point out where they arrived to the similar conclusion, these two sources to explain the genetic diversity present in the Americas. But the researcher found a signal of yet a third population, a third group in individuals, really isolated groups from Amazonia. This population are Caritiana, Surui, and Chavanti. And those groups, on top of these two ancestry, the let's say Siberian ancestry and the East Asian ancestry, they have a third component that is shared with populations that are in Austro-Melanesia, so for example Papuan or Ongi, and it has been named population Y. It so far is still not clear how this genetic component arrived and survived through time in this isolated place. So the oldest genome ever published so far from the Americas is this Clovis genome. It's called ANSIC1, and it's a Clovis-associated genomes. Who are those Clovis? Clovis are among the first population or groups present in North America, and they are characterized to be really successful megafaunal hunters, and they are typologically they are characterized by these points that are called Clovis points. And what was really exciting about this paper published in 2014 was that when the genome of this ancient individual was sequenced and compared to modern day individuals from all around the world, it shows the highest genetic similarities to people living today in Central and South America. So placing this genome on this southern branch of expansion into the Americas. Last year was kind of a prolific year for ancient DNA from the Americas, because we finally got more genome from North America, and this publication in Science showed that they could sequence genome both from the coast, the Californian coast, as well as Mexico, and then also from north and southern southern Ontario, and they could show that these two groups, these individuals fall in two distinct groups, and they actually resemble this early diversification that I was telling you at the beginning between Northern Americans and Central and South Americans. Just to be more neutral in terms of where let's say about the geographical location where the split happened, we just described them as, we just called them ancestral B for the one that, this group that left traces in Northern American population, and ancestral A for people that left traces in modern day people from Central and South America. So we'll talk about ancestral A, ancestral B later on. So as you can see here from this map, unfortunately, before, by the time of this publication, there was almost no ancient genome published from South America. So we tried to start, and we started a project. We already started a project to actually try to fill these gaps, and with three main questions in our head. We wanted to understand if Central and South American variation of early in the past mirror the population structure that we see today. So if there is some genetic continuity through time, we want to understand what is the minimum number of genetic exchange between North and South America. That means how many waves we have from the north to the south. And then we want to, of course, try to reconstruct the relationship between different groups in Central and South America through space and through time. So, and this was the data set that we assembled last year. It's 14 new genomes, sorry, not 14, 49, it would be a little bit less. 49 ancient individuals spanning between 11,000 and 600 BP. But as you can see from our timeline, from the oldest, that is ANSIC, the Clovis-related genome, to the youngest, 35 of the new ancient genome that we report are actually older than 3,000 BP. So we really try to understand what is the diversity of the initial settlement of Central and South America. And those are divided into four, let's say, geographical transect. We have individual from Belize, Another transect is from the central Andes. Then the third cluster is from individual from the southern cone, so from Chile and Argentina. And finally, we have some individual from the southeast Brazil. And this map here that you see is not only reporting the new ancient individuals, but also is representing a meaningful statistic. And probably you cannot see really well, but the different colors of these dots and squares represent the affinity of those individuals compared to Clovis. So we wanted to test if every individual in Central and South America is equally related to the oldest genome, the oldest North American genome, that is Ancien, or there are differential relationships. And what we were really surprised to see is that actually the two oldest genomes that we sequence from South America, that there is that are population from a group of individuals from Lapo Santo dated to around 10,000 years ago, and one single individual from in Chile in a site called Los Riles dated to 11,000 years ago. So the oldest genome show a higher affinity to Clovis. So the oldest genome show a highest affinity with the oldest from North America. So we were really interested to study that further. So what we did, first of all, is like typical PCA, principal component analysis, to distinguish population in the genetic map. And what we identified clearly is these two clusters, ancestral B, North America, and ancestral A, that contribute to Central and South America. However, if you see where our ancient genome from South America fall, they're all on top of each other. We were not really able to distinguish that based on this principal component analysis. So we decided to use a different technique, that is this F3 outgroup statistic represented through MDS plot. It stays for multi-dimensional scaling plot, and this is actually nothing else than a matrix that measures the dissimilarities between different groups. And what emerged from this analysis is that we could actually group ancient individuals based on their geography. So we could identify a group of individuals both very ancient, but also more recent, from Fuegians, from Fuego Patagonia, forming one cluster. Then we have both ancient and modern individuals from Andes forming another cluster. And then the Brazilian individuals again falling together. And then we have this Belize individual kind of in between. So what we try to do is like, okay, the ultimate goal of this exercise is see if we can reconstruct a kind of a tree, a graph that can represent all these individuals together in a phylogenetic manner. And so we start to kind of build this so-called QP graph of early Americans. And after the formation of the source population of all Native Americans, that, as I told you before, is a mix between Han Chinese and these Siberian ancient hunter-gatherer. There is a first split of ancient Beringian, a genome found in Alaska. And then, again, this really quick diversification that happened probably around, before 15,000 years ago in North America between this ancestral B and ancestral A. And what was really interesting to see is that within the ancestral A lineage, all the diversity of the ancient Central and South America fall. So all ancient Central and South America are part of this ancestral A lineage. And what you could notice really clearly is that a genome from different locations, from the Andes, from the southern cone, from Belize, and from Brazil, they kind of diversify extremely fast with a sort of star-like diversification. Meaning that, or we could speculate that really people arrived in South America and spread and diversified extremely fast, so that they are like different branches on a star-like diversification. The problem starts, and it becomes much more complicated when we include in this admixture graph Clovis, when we add the oldest genome from North America. And so what we see when we do this exercise, and we add Clovis, Ancien, we see that it becomes like a spaghetti kind of a tree. But there are certain characteristics that they're shared with the previous graph. That is, of course, this diversification, early diversification between ancestral B and ancestral A. There is, again, this star-like radiation of South American individuals. But what is interesting, we identify 2 additional gene flow from North to South America. One gene flow is this one in light blue, is a gene flow of a specific affinity from Clovis into the oldest genome from South America that are, as I told you before, Chiles losriles 11,000 years ago, as well as Lapadosanto 10,000 years ago. But this is not the only signal that we find, not the only gene flow that we find. There is an additional one that appears much later in individuals from Peru, at least from 4,200 years ago, from a group related to the California Channel Islands. So if we try to sum up a little bit what I just showed you before in a kind of graphical abstract, we identified a total of 4 genetics exchanges between North and South America. One is a primary source of ancestral A ancestry in all South Americans, at least by 9,000 years ago. Then you have again a new lineage with distinct affinity to Ancien, and that we find in the oldest individuals from Lapadosanto and Los Riles, and then yet a third gene flow event that happens much later, around at least by 4,200 years ago, and we link to population from the California Channel Islands. And of course, the population Y signal is found in modern-day Amazonians that we could not find in any of our ancient samples. But of course, as you can see, we don't have samples from Amazonia. So I will skip on these slides, just to tell you that we are doing much more work now on the Caribbean, but I'm happy to talk with you about that later. And I would like to just conclude with these last slides about what are the next steps. And we finished the paper in 2018 by writing from the period between 11,000 and 3,000, that includes most of our individuals. We lacked ancient DNA data from Amazonia, North and South America, and the Caribbean, and thus we cannot determine how individuals from this region relate to the ones we analyzed. So what we're actually doing today is try to expand the collection of ancient genomes from Northwest and South America. We are really interested also to get more data from the Circum-Caribbean region. And of course, one of the reasons why we are here, I'm really interested to talk to you, is also establish collaboration with Cuban researchers that share the same interest, and potentially we can study the population history of Colombia and the South American together. And with that, I would like to acknowledge the group in Jena, and especially a PhD student, Catherine Negele, that is actually worked a lot in this, on this topic. And you for your attention.
Thanks, Dr. Post. Now it's time for Dr. Andrea Casas Vargas. She's from the Instituto de Genética y Departamento de Antropología de la Universidad Nacional de Colombia, and she's presenting on the complete mitogenome of ancient human bones from the northeastern Andes in South America. Dr. Andrea?
Good afternoon, everyone, and thank you for inviting me. Well, this project was carried out in collaboration with the Universidad Nacional de Colombia, the Pontificia Universidad Javeriana, the Institute for Legal Medicine of Innsbruck, and it was grant sponsored by Ciencia. Here, I will be talking about topics related to the peopling of the Americas by anthropological perspective. In this context, two specific variables, the shape of the skull and the dental size, are considered. As for the shape of the skull, we have the dolichocephalic type that are elongated skulls, characteristic of Paleoamericans, and the brachycephalic type that has a more rounded shape and is characteristic for recent settlers. In terms of dental size, the classification is divided into macrodonts, which are large teeth, and microdonts, which are small. Different peopling hypotheses, such as migrationism and microevolutionism, have been formulated based on these two variables. These two differences are portrayed by the oldest Paleoamerican throughout the American continent. This map here shows the geographical location of some bones remains with Paleoamerican morphology. One of Colombia's most studied archaeological sites is found in the Eastern Andes. Some of the most found remains in our country have both types of morphology. The oldest remains show a dolichocephalic morphology, while the ones from posterior periods have a brachycephalic morphology. Based on these findings, it has been to classify these remains in different archaeological periods. The early Preceramic period is characterized by dolichocephalic morphology and large teeth. Individuals from this time were hunter-gatherers, and the archaeological sites are Chequa, Tequendama, Nemocón, and Floresta. The late Preceramic period is also characterized by a dolichocephalic morphology, but with small teeth. Here, hunting, gathering, fishing, and horticulture were the predominating activities. Remains from this time have been found in Aguasuque and Vistahermosa. The next period is the formative or Herrera, in which the brachycephalic type, morphology, and cranial deformation appear for the first time. During this period, people started farming and congregating around a small village, and inside pottery can be found. The archaeological sites are Madrid, Laguna de la Herrera, Sogamoso, and Duitama. The early Mooskaa period is characterized by brachycephalic morphology. This is considered a transition period in which the painting ceramics also appear. The archaeological sites are Candelaria, Porto Alegre, and Sogamoso. Finally, in terms of the late Mooskaa period, there are brachycephalic morphology, intensive agricultural activities, and the societies became hierarchical. Remains of this time have been found in Bogotá, Tunja, Duitama, and Sogamoso. Considering these findings, it is important to begin to enrich hypotheses about the peopling of Colombia with the result obtained using genetic tools. One of the most useful genetic tools for reconstructing ancestry is the mitochondrial DNA, and there are four main reasons. High copy number, which means that large amount of mitochondrial DNA can be obtained. Maternal inheritance, which provides a more direct genetic lineage. It is not recombining, and it has a high mutation rate. Based on these results from mitochondrial DNA studies, it has been possible to map the world's human peopling pathways. These pathways are represented by continent-specific haplogroups. In the case of the American continent, 5 main haplogroups have been found: A, B, C, D, and X. A region that allows us to perform both temporal and spatial population genetic analysis is that of the Sun Temple in Sogamoso, as it is located 210 kilometers from Bogotá. This site was a religious center of major importance for the Muiscas. In this place, bones from all 4 archaeological periods have been found. Bone remains belonging to the pre-ceramic period dating from around 8,000 years before present. Remains from the formative period dating from around 2,000 years before present, and remains from the early and late Muisca period. These findings have made the region ideal for temporal spatial analysis. For this reason, our main purpose was to analyze ancient skeletal remains from 3 consecutive cultural periods found in the surrounding of the Sun Temple, to shed light on their population dynamics and origins. To complete this purpose, we worked in collaboration with Dr. Walter Parson and the Institute for Legal Medicine of Innsbruck, and concluded the sequencing of the complete mitochondrial genome. We analyzed a total of 45 samples belonging to the late Muisca, early Muisca, formative, and pre-ceramic periods. Mitochondrial haplogroups were defined, and we obtained these frequency distributions. In the Muisca period, we found haplogroups A2, B2D, C1b, and D4H3A. Within the most prevalent haplogroups A2, subhaplogroups A2AC, A2AD, and A2Y were found. In the formative period, we found haplogroups A2AC and B2D. In the pre-ceramic period, was obtained results for 2 individuals, one with C1b and the other one with C1b8. A network analysis was performed based on information from the mitochondrial DNA control region, which showed that there are some shared haplotypes. In haplogroup B2D, there are shared haplotypes between the formative and Muisca periods. And in haplogroup C1b, there are shared haplotypes between the pre-ceramic and the Muisca period. A comparison with the other population was performed. This comparison showed haplogroup A2AC as the most frequent in our sample, and it is highly present in modern Colombian populations, including the Embera Chamí, Vaupés, and Bogotá. This haplogroup is also shared with population from Panama, Venezuela, and to a lesser extent from Ecuador and Peru. Haplogroup A2Y had previously been reported in an isolated Waurani population from Ecuador and was thought to be unique to them. Another important haplogroup is A2AD, which has 3 clearly differentiated clusters. The last cluster is characterized by a polymorphism in the 60,259 region that led us to consider it a new lineage. This haplogroup has also been found in contemporary population from Panama and the Dominican Republic. In this phylogenetic analysis, our samples from the Muisca period are shown in red boxes, and samples from the formative period are shown in orange boxes. We found that 8 of these samples belong to the A2AC1 lineage and form a structured subclade characterized by a transition in the 9,591A region shared with 2 contemporary mitogenomes from Colombia and Bogota, and Cuba. These findings leads us to consider this subclade as a new lineage. The coalescent age for the new A2AC1 calculated as 4,000 years. Additionally, in haplogroup A2AD, there is evidence of the new A2AD lineage, and the coalescence age for this new subclade was almost 10,000 years. One complete mitogenome from this study, belonging to the Mooskaa period, shared with two extant Ecuadorian Waurani haplotypes that had defined haplogroup A2Y. The coalescent age for this lineage was calculated as 10,000 years. In terms of haplogroup B2, when analyzing sublineage B2D, we found that two mitogenomes from this study, one from the formative and one from the Mooskaa period, were identical with 5 modern Colombian samples. The coalescent age of B2D was calculated to be 7,000 years. Haplogroup C1b was the only complete mitogenome from this study. This haplogroup belonged to the Mooskaa period and could not be assigned to any described subclade. Haplogroup D4H3A from this study, also from the Mooskaa period, fell into an unnamed clade defined by two transitions shared with two extant samples from Mexico and another sample whose origin is unknown. Dispersion analysis of mitochondrial haplogroups from the MPOP database confirms the high frequency of haplogroup A2AC in northeastern South America. Shows that haplogroup A2AD is scattered in Central America. Unpresent lineage A2Y as restricted to an isolated population in Ecuador. On the other hand, haplogroup B2D is shown in two focal points, one in Central America and the other one in Colombia. Lineage C1b is currently spread throughout the continent, and C1b8 has been reported in California, Mexico, Venezuela, Ecuador, Chile, Argentina, and Brazil. Finally, D4H3A is a rare founder haplogroup, first described in Ecuador, with currently mainly spread along the Pacific coast in South America. In conclusion, this first complete mitogenomic report of pre-Columbian human samples from Colombia demonstrate how ancient DNA samples can contribute to the reconstruction of a more complete picture of the past. The novel ancient mitogenome from this study, belonging to all 4 major clades of Native American founders' mitochondrial DNA lineages, had highly desired data for reconstruction details of human migrations. Unlike other ancient mitogenome studies, we reported polymorphisms beyond the haplogroup nodes, thus contributing to more precise phylogenetics and convergence estimates. These samples set clearly shows, at least for the maternal side, that despite European conquest, the genetic heritage from the original human settlers of the Mooskaa territory is preserved in today's population at a high degree. Finally, I thank you. Thank you for your attention, and thanks to all research partners and institutions. Thank you.
Thanks, Dr. Casas Vargas. Well, we have some time for questions. I would like to ask the first question to Dr. Cosimo and Andrea. The thing is that we have here an old hypothesis about the contribution of people from the Pacific Islands to the ancient populations in South America. What about that?
Maybe I start on that. Yeah. When it was first discovered, this signal in the Amazonian population from today, people thought, okay, that's actually the Polynesians. But looking more in depth, this ancestry does not correspond to Polynesian ancestry. And actually, there was a couple of studies, maybe you are aware of, where they sequenced the genome of Botocudo individual from Brazil, or at least found in Brazil, and they turned out to be full Polynesians. And later on was discovered there was a sample mix-up, actually, where this skull were labeled Botocudo, but were brought to Rio de Janeiro during an exhibition a few years few hundred, tens of years back. So far we do not see any evidence of a contribution for Polynesian population into the gene pool of Native Americans. Yeah.
Somebody else? Over there, at the end?
So I have two questions for the same speaker. The first one is if you have any sociological or biogeographic or historical reason why the other group of Native Americans, like from Northern America, never migrated or mixed up with the other subgroup of Native Americans.
No, actually, I wouldn't know the answer. It's a good question, but I can just show that contrary to what was proposed in the first study, 2018, where they first reported those genome, we did many more analysis on that, and we do not find contribution for this ancestral B, what we call, into South America. So probably, you know, they were occupying a different geographical region, and you know, they show genetic continuity only in the north, but not contribution in the south.
And the other is if you see any consistent genetic difference between modern and ancestral South Americans, like some selection in some region of the genome or something like that.
Can you repeat, please?
If there is any consistent difference genetically between modern and ancestral South Americans.
Yeah, I mean, that's a really $1 million question. I mean, we are really interested to study selection. However, with the kind of data we are looking at, we are looking only at population history. We usually target position in the genome that are not under selection. We try to target the so-called neutral loci. But of course, with this information, with genome-wide information, we can also study selection and see, you know, how people, for example, respond to living at high elevation, and if there was a selection in the genome. But so far, these studies are really starting now to happen, because the main issue is that we have so little data to do this kind of, you know, selection scans.
Another question. I was curious about the change in the shape of the skull, because it's quite dramatic. Is it associated with, what could be the reason for that? Is it migration from other populations? Is it a change in the diet? What could be the reason?
This hypothesis is possible to variables. Or migrationism is the first hypothesis, or microevolutionism is sorry, I need to translate to Spanish. Bueno, casi todos son españoles, ¿no? Pues las hipótesis que hay a nivel arqueológico se basan entre las dos, si son migración, si fue a partir de rutas migratorias que reemplazaron otros grupos y por eso hubo un cambio morfológico, ¿sí? Es decir, que cambiaron, que llegaron primero una oleada migratoria de tipo dolicocéfalos y llegaron una nueva oleada migratoria con la morfología braquicéfalo, ¿sí? Hubo un reemplazo en cuanto a la morfología. Pero hay otros estudios que también a nivel de microevolucionismo, quiere decir que por diferentes eventos evolutivos, microevolutivos in situ, hubo esos cambios, provocaron esos cambios y muchas veces acá en Colombia tenemos la fortuna de ver como esas diferentes temporalidades de los precerámicos y ya de los agricultores. Entonces, al ver esas transiciones de morfología, se ha hablado mucho del apoyo más a la hipótesis de microevolucionismo, ¿sí? Que más es por diferentes efectos de dieta, ambientales, diferentes factores externos llevaron a esos cambios morfológicos.
You like to translate?
Sí, me ayudas a dar. I don't know.
He's translating over there? Okay. And I will use another one. The shape in the school conformation, is it shared by men and women, or is it more common in any of the sexes?
No, it's a both,
Okay,
really.
good.
Se han encontrado los dos. Sí, se han encontrado tanto en mujeres como en hombres. Pero a veces estos restos son más escasos, entonces hay uno o dos individuos, entonces normalmente se encuentra a veces un solo un hombre, pero también se ha observado también en infantes, en babies.
Yeah, if I can add something about your explanation about these Paleo-American features, at least what we can say is that there is not an additional migration from outside of the Americas bringing this different morphology. That was proposed in the past, for example, by Walter Neves, about this different ancestry coming into the Americas, bringing different morphology. This one we can exclude, but of course the two questions are still open.
Thank you. Thank you all.
Buenas tardes, good afternoon. We have arrived to the last part of this meeting, and we have seen talks about very big systems, as for example, the sun, our whole planet, big ecosystems, as well as very small systems related with neurosciences as well as with genetics. And we have seen both natural and social systems, including what happens with the human laws. And behind most of these talks, there is a word which has been present or absent, which has to do with complexity and how these systems get organized. So for this part, we are going to be talking about linguistics. And linguistics is in the realm of complexity. And it is so because it is the first step into structuralism. And understanding the language means also to understand many things of human beings. Ferdinand de Saussure used to say that, or it was quoted to say that if you want to understand man, or human beings, excuse me, you have to get to what is exclusive to human beings, and it is language. Myself, and my name is Rafael Hurtado, I'm a physicist, and it is incredible that linguists and physicists, we are very, very close together because we look at nature at the smallest or more elementary parts of them, their constituents, as well as how they are organized. A very important physicist for our community, Murray Hellman, who is responsible for posting the hypothesis of quarks content of matter. After he, I don't know if he got tired of particle physics, he went into linguistics, and also he founded the Santa Fe Institute for Complexity. So now we are going to start with Dr. Matthijs List. He comes from the Department of Linguistics and Cultural Evolution from the Max Planck Institute for the Science of Human History. We have had already 2 talks from representatives of this institute. And his talk is going to be on the search for the drivers of linguistic and cultural diversity.
Yeah, many thanks for the very profound introduction into linguistics. And as we are talking all the time, it's important that we have a talk on linguistics, right? Because, but although the problem of linguists is that the same problem that maybe doctors have. If you have a doctor and you sit on the table in the evening with some people that you don't know, they will just tell you everything about the diseases that they have. At least it happens to my father often, who is an orthopedist. So it's not the worst diseases that the people talk about. But in linguistics, the problem is everybody talks. So everybody thinks they can talk and think they understand language. But it's actually embarrassing if we think how few things we, the human beings, the scientists, understand about the things that we use in order to transport science and to discuss about it. We don't intend even to solve these questions at the Max Planck Institute for the Science of Human History in our department. We want to know where it comes from and how it developed. So we know that this is a really challenging task. Finding the drivers of cultural and linguistic diversity is something that we may never really know, but that makes the task also so interesting. The most interesting part about languages, however, is, or why can we actually use languages in order to learn something about the past? Why? Is because languages are similar to fingerprints, to to bones, similar to languages are to DNA. They are keys to the past. And this has been known already for a long time. 200 years ago, like Jakob Grimm and Rasmus Rast, they detected that languages that are spoken, like Icelandic, Latin, Old Greek, and Sanskrit, that are spoken in very different regions of the world, actually go back to the same ancestral language. And how did they find it out? They used a method for language comparison. They developed a new method for language comparison that is based on the intensive comparison of individual languages, and that is based on identifying regularly recurrent similarities among these languages, and that is then used to prove that the languages are in fact related and then have spread from a common source. And this method has not only been leading to the development, to the detection of the Indo-European language family that is like spread like this, and now we have it even in more places of the world, because here we speak Spanish. Más despacio. ¿Por qué? Okay, now we slow down, but for what reason? Do I have only... Are you... Ah, okay. Lo siento. Okay, recomenzamos. So this family, so the comparative method, as the people call it, yeah, let's go back. The comparative method, the method for language comparison that is called comparative method, has led to not only to the development and to the detection of the Indo-European language family that we can see here, and is nowadays spoken in almost all places of the world. Like, I'm speaking an Indo-European language here. If I spoke Spanish, I would also speak an Indo-European language, yeah? But we have detected many, many more language families on Earth. At the moment, we count more than 300 different language families, and it depends. Some people even count more, some people count less. The problem, however, is that despite these methods and despite 200 years of research, as we have been doing already, our knowledge about all these language families is still rather fuzzy. And the problem is based on the method that we use, because this method is carried out manually most of the time, and it makes it very time-consuming. You need to be a real expert to be able to apply the method. You need to know the languages very well. And this also leads to certain problems with consistency, because there are no real formal guidelines. So I could not say what is the alphabet, the ABC of the comparative method here, because we lack these formal guidelines. Also difficult to evaluate actually the different findings that people come up with. As an alternative, people have been proposing automated methods that would just replace the experts, like similar to the way that we are trying to replace people in wherever we can, because it's cheaper. We have tried to replace the linguists. So this is of course not completely honest what I say here, but there was attempts to compute the task and to formalize the task, because we all know also that this is important. This does not, as you will see in a minute, mean that we want to replace experts. But the problems of the computational approaches, they have been developed for the last 20 years or so, and I myself have been doing some work in this domain, is that they lack flexibility. Usually you can only apply them to one specific task, maybe sometimes even only to one language family, so this is actually boring. We want to apply them to all the languages, to all the problems. They also lack accuracy, because usually the humans are still better. Yeah, it's not like chess, where the machines are already better than the humans. This is a different task. So if the people really want to show that artificial intelligence is better, is getting better than the humans, then prove it on languages, please. They also often rely on manually annotated data, so the people will still use the manually annotated data in order to actually analyze the data afterwards with help of computers. And sometimes they produce results in a black box fashion, so we do not really know why the algorithm will turn out a certain result, and actually that is not convenient, right? Now, the problem that we have is that the linguists have been sitting in the ivory tower, yeah? For a long time, for centuries, and they have been enjoying that, yeah, because it's cool, it's a nice climate there inside and everything. And now all of a sudden comes the storm of bits and bytes of the computational linguists that I represent to some part as well, and the people were afraid what will happen with the ivory tower of historical linguistics. So would it crash it? Would it tear it apart like the storm of bits of byte? Or maybe the alternative, would it help us so that the tower shines in a new glance? So the answer that I can give to that is actually, and this is what the project, what's also the agenda, a great part of the agenda at our Department of Linguistic and Cultural Evolution is about, is that we want to combine the two profiles. So we have the geek on the one hand, so the computer scientist that is only interested in bits and bytes, and we have the classical linguist here that has a good ideas, actually knows the languages very well, and reads books with very long titles. We want to combine them and have them work together closely. And we think that this will actually bring some new spirit into the whole discipline and will help us to find new things about the drivers of linguistic and cultural diversity, but also of other aspects of linguistics in general, but maybe even about cognition and about other questions. So what do we do here, actually, as an agenda? We have a strategy, and I will briefly introduce all the three parts here. Our strategy is to lift the data that has been accumulated already over the past of languages and cultures from analogous to digital and from digital to comparable data. We want to integrate classical research with modern computational approaches and research on linguistic and cultural evolution with research in archaeology and archaeogenetics. We want to ask questions about the concrete past of our languages and cultures and the drivers of linguistic and cultural diversity. And depending on the time I have, I will now start telling about that. So the first part is lifting the data. This might seem easy. People sometimes think that there's no problem with the data, but the data is usually manually annotated. Usually, if we want to lift the data is there in raw form. So we have some sheets of paper, maybe even some sound files, maybe people, sometimes only people that say that they speak a language, or they say they have been speaking with people who speak a language. So we lift it in the first stage. Usually people have been already lifting the data to a stage of analogous data, so you find it in form of books, big books that present you something about the grammar of a language. But here we need to go, of course, to the digital form, but the digital form is not everything, because digital data can also be messy and heavily incomparable, and that is what we usually find. So we need the next stage, and the next stage is what we call, following Wilkinson et al., fair data. Fair data is comparable data. That is data of languages that people produce that can be compared directly. So we can compare data for one language directly with the data for another language. Fair means here findable, accessible, interoperable, and reusable. So it should be it should be findable, it should be online, it should be easy to find the data. If you want to know something about a language, you need to be able to find it. And you should be able to find it easily if the data is online there, out there, somewhere. It should be accessible. There should be no paywalls on it. It should be open data, open research. It should be interoperable. So if you download the data, it should be in a format that you can use, not something that is like completely unusable for you. So you want to use it in different, maybe algorithms also, because our work heavily relies on algorithmic approaches. And it should be reusable. So you should be able to then actually reuse it for other tasks than has been compiled for in the first instance. And at the moment, the data is not like that. Most of the data in historical linguistics or in linguistics in general is not fair in this sense. We have created standards that we call cross-linguistic data formats in order to account for what we think is fair in terms of data that we can compare. These are like words pronounced and the pronunciations of certain words that mean certain concepts in a certain language. Another thing, so you can think of a lexicon or a word list of different languages, but we also have data on other aspects. So these standards, as I tried to depict here with a banana and a lineal, you can see these standards, they are using very simple formats. We use spreadsheet formats, so we don't want to use very complex things, databases or what other things. We have validation software, so with the software you can see whether the data is actually in the format that the people claim that it is in there. And we have also specific things that we call reference catalogs, and they are somehow, the rendering is not really as I wanted it to be, but reference catalogs are catalogs that tell you something about where the points comes from. For example, Glottolog has an ID identifiers for language names, glottolog.org. Glottolog will tell you something about where the language is spoken, how many speakers there are, which literature describes the language, and will also give you some authoritative ID identifier of 8 symbols, 4 numbers and 4 letters, so that you can actually say, I talk about this language. Because often linguists use different names for languages, and this leads to a lot of confusion whenever linguists talk, because you don't know about what language they speak or whether they speak about the same language. First point is standardization. Second point is with Concepticon. Same thing for concepts. So you want to talk about apples, yes, then talk about apples, but tell us what apples you talk about. We talk about the apples with the ID 561, for example. The Concepticon is a catalog of more than 3,000 different concepts that are important for languages to describe a language. Not necessarily the concepts you want to use when you go as a tourist guide and want to talk with other people in another country and want to learn a language, but the concepts that reflect the core, the development of a language, and have been used by linguists for the last 200 years already. So this is a very big catalog that currently links more than 250 different sources and explains and tries to define the concepts. And then we have what you cannot see here is cross-linguistic transcription systems, a same system for sounds. So if you want to talk about a p, then we have an identifier and a complete description for that. But if you use another sound that is slightly different, like a b, then there is another identifier, and we have a transcription system. You might think that this had been already solved in the past with the International Phonetic Alphabet, but this is not the case, because the people don't use it in a standardized form. So we use all these standardized efforts, and once you have those, you can make an easy online publication of the data and share it and make the data findable in the CLLD framework, which stands for Cross-Linguistically Linked Data. Examples for the data that we have lifted so far is Dplace, a big global database of cultural, linguistic, and environmental diversity. Another example is GramBank, database on grammatical structures, 195 structural linguistic features for more than 1,300 languages at the moment, spanning 260 language families. And we have, these two are still work in progress. LexiBank, a repository where we want people to give the chance that they can actually deposit their data after they have made a publication, and this will aggregate the data in the fair form so we can accumulate the data similar to GeneBank in biology. So we have culture, grammar, and lexicon. We have it more or less covered, but the question is, of course, what do we do with the data once we have lifted the data? We integrate, for example, with computational methods. I myself have been working on computational automatic word comparison with a LingPy package. LingPy stands for Linguistic Python on lingpy.org. It's a Python library that you can use in order to compare words automatically. At the moment, it's a state of the art. Over 50 publications based on LingPy. By now, it's already over 70. Multiple you might think that it's not a lot, but we are talking about humanities here, so. Multiple phonetic alignments, they are like algorithms for specific parts, automatic cognate detection, correspondence to the pattern identification. This has already been published, actually. Last time I used this slide, it was still under review. And the library has a high accuracy, so if you go for the phonetic alignments, it has something like 98% to what experts have sometimes. So this is a task that is also considerably easy, you could say. Other tasks, it is not that good, for example, finding related words, what we call cognate detection. There it's like, reaches something like 89% on average, depending also on the time depth of the language families. And we have other parts where we actually get sometimes quite good and sometimes less good results, but it's something that we use now in order to compare the data automatically, and it helps us a lot. Another example is we have been working on cross-linguistic collexifications. That is something like words that are polysemous across the languages in the world. So many languages don't use, for example, for words like forest, tree, wood, stem, branch, and root, you may often use the same word in the languages. You can see that, for example, in French, where you say bois, for both forest and for wood. You can see it in English, where sometimes you also say in the woods and the wood, right? Where you actually, and we call that co-lexification, yeah? So you co-lexify two concepts. If you do that, you look at different languages here, Russian, direva, we can make a network out of those. And if we have these networks, they reflect in some sense, cross-linguistically, cross-culturally, they reflect human perception on a very rudimentary level, because there is usually, when people do that actually, when we have the same word for two different concepts in a language, this means the concepts are similar. And if you then use lots of data for that purpose and compare those patterns across the languages in the world, you find the patterns that are at the core of the cognition that I would say actually they open a window to the human cognition from a perspective that has been completely forgotten or has been completely underexplored up to now. Here you see the sample of all the languages that we have lifted so far, more than 2,300 or even more, 2,893 varieties, sometimes the same languages, dialects, etc. 2,809 concepts, 200 language families, and 30 basic data sets. This is an impressive collection, and if you look at this connections between the concepts that we have in this data set, you see it's a small world network. It's like larger than my slides, and everything is connected, which is actually quite interesting. But if you look at the details of the connections with specific algorithms, you can see that the connectivity itself gives you some things that make sense. So you can see that the knee and elbow are connected. Why are they connected? Because they are similar, right? If you actually looked at, but not all the languages in the world would say that knee and elbow should be given the same word. You find many things that are there reflected, and it's really a really rich resource for people who are interested in the human mind or in the way that we perceive the world, also in different cultures or cross-culturally, by just looking at this from different languages. Now, interdisciplinary research. Cosimo has not talked about this, but because he was concentrating on the South America case, but they had another very interesting paper that was also last year coming out, in which actually there was a collaboration between our department and the Department of Archaeogenetics, where they found out that despite population replacement, there was language continuity in remote Oceania. And this was based on This paper was based on a field work station that is currently run both by the archaeogeneticists and the linguists of the Institute for the Science of Human History. I myself was involved in a project with where we tried to look at archaeology and archaeological findings as they are already given in the literature, and we just looked at the linguistic data ourselves with colleagues from Paris, where we looked into the origins of Sino-Tibetan languages, and we found out that the most likely route of expansion would start here from the Yangshao culture and then go here, because we find the words only reflected in most of the languages already here. So the word for pig is very similar across all the languages, so they brought it with them, but other words were only acquired later on. So this is the evidence that we used in order to draw our conclusion, and also say like the Chinese, in this case, it's about 7,200 years old. That is our guess. We don't know. We will see in a couple of years whether new people come out with new data. Another part, but this is not research that I can publish, is about poetry that I have published so far, but it's actually poetry has three different dimensions. So poetry is based on the human brain because we need to speak, we need our brain to speak, so certain things are all the same among all humans, I would say, right? Poetry is also based on a language. If you have a specific language, I mean, in some languages people don't want to rhyme because it's too easy or because it's too boring. In some languages they only want to rhyme because everything else is too heavy or we don't know. It's like all these patterns, some languages are more rhythm-based, so poetry has this aspect, and we have culture, of course. Sometimes you just perpetuate a certain pattern because your culture imposes you to do so. These three, actually, the interaction of language and culture, which looks a little bit ugly here, is very interesting from the evolutionary perspective because you can actually look at how these change through time. But in order to do that, you must first actually, so here's again like they are the three C's, yeah, cognition, communication, and culture. But if you want to see, and Roman Jakobson, the famous linguist, was actually talking about that there is a specific poetic function in language. So when we want to understand language in general, we also need to understand the poetic aspect of language, which actually shows how important it is that we look at these patterns. Now, what we started is doing an annotation framework for poetry that allows us to annotate on a very detailed level how, for example, specific languages rhyme. This is an example from a German poem, which gives us all the information on the rhyme words here. So we have Dämmerung will die Flügel spreiten, schaurig rühren sich die Bäume, Wolken ziehen wie schwere Träume, was will dieses Grauen bedeuten? What you can already see here is that the Germans have a weird way of rhyming because we rhyme spreiten with bedeuten. Not many languages follow this pattern that that they allow to rhyme I with oi. Here's a Russian example. Leta. Я Жан как лето, время есть, а денег нету, но мне на это наплевать. Лето я купил себе газету, газета есть, а пива нету, я иду его искать. Another example. We have a Chinese example. 你问我爱你有多深，我爱你有几分，我的情也真，我的爱也真。月亮代表我的心, that is talking about the moon, that expresses what I think. And we have Bob Dylan. I want to have a big collection of all the poems in the world, and then we can analyze them automatically because we are working on all these methods and try to actually expand them, etc. And this is actually what I really hope that we can do in the future. And we have a Spanish case. Actually, I don't know, you need to correct me, but actually I think that Spanish is really weird in terms of rhyming because there's things like you can rhyme amor with canción, as in this song from Silvio Rodriguez. But maybe it's just that I'm just not giving the right analysis here. So we need to collaborate on that, and then we can actually find out more about the evolution, the history of our cultures. Here's a rhyme network that we made, and again we have a small network word. This is Chinese poetry from 2,000 years ago, and amazingly, all the words that rhyme with each other, so they are all connected at some point. You can find the patterns of how they were pronounced, but they rhyme. So it's rather messy what we have here. And the question is, are all languages that messy, or are they more strict in the poetry that they have? Asking questions. I have been asking many questions already. Maybe you have been asked. But what we want to do is also, and what is actually one of the great advantages of interdisciplinary research is that you are actually sometimes you are forced to be confronted with questions that you would never ask. Linguists are really good in not asking questions. For example, also almost 200 years ago, in 1871, okay, I'm not good in counting, 150 years ago, the Société de Linguistique de Paris decided that they would never talk about the origin of our languages again. And since then, linguists have more or less been doing that. So you learn that in university when you start in your first course. No, we don't talk about language origins. And now people talk about it again. So the taboo is broken, and it is actually many interesting questions that we can solve with this, but we need to ask the questions, and this is only thanks to interdisciplinary research that outsiders came in and said like, sorry, but why do you do it this way? Is it really important? So we need sometimes the questions, linguists would think like, why is it such a naive question? I'm not interested in the origin of language, but it's interesting, right? I personally am not interested, I have to admit, but because I was raised in the old fashion, yeah? Anyway. Particular questions. For example, we could ask how old are the many different language families in South America? Cosimo told me yesterday that apparently the genetic diversity is really, really close. Yeah, the languages in South America, like the populations are genetically really close, but linguistically they are the most diverse place in linguistic diversity on Earth, almost. So it's really incredible how we have linguistic diversity, but like genetic diversity is less. This is an interesting question, actually. How did the language families of the world spread through their history? Also concrete, so as we try to do for the Chinese languages. How did the new concepts and new structures spread through the sociolinguistic networks of Southern American languages? Sometimes it's also interesting to look at shallow history and see actually how new concepts influence and spread, because then you can see how the people actually interact with each other if you don't have other evidence. So you can use linguistics for many aspects by investigating language and actually use that in order to infer many more things about the past, about the human past, about the past of populations, etc. And we also need to ask general questions. What are the most general cross-linguistic tendencies of sound change? Sound change mostly regulated within the system of a language, and it's amazing how regular that is. We have made an experiment with a colleague where we predicted which words are pronounced in a certain language that he's doing fieldwork on, and he forgot to ask certain words in the first part. So now he went back and we predicted the words, and we predicted them with more than 75% of regularity. So he would actually have not needed to go there. Of course, he needed to go there in order to confirm it, but we can predict how a language pronounces a certain word if we have enough evidence already about the history and the patterns and the structural patterns in the language. What are the most general cross-linguistic tendencies of semantic change? Also something. Sometimes we have seen in a network that we can see some languages say like knee and elbow are similar, but there are many other things that you can actually perceive as similar. But what is similarity generally? This is a similarity that is on the surface, but sometimes it's also continuity. There are many things also that relate to cognition that we need to understand more properly. And also are there general tendencies of word exchange and spread, of borrowing as we call it, but it's not borrowing, because if you borrow a word you never give it back actually, so it's more like stealing, as David Morrison, a colleague from biology, told me once. So are there general tendencies of that? Maybe not, because maybe you only borrow the words of the technologies that you don't have if you are in a certain population, but maybe there are. So these are all interesting questions that we should ask. And as an outlook, I just want to show like where we are currently working on. So we have been working on Sino-Tibetan so far, but we are also already working on other parts of the world. So we try to shed light on Sino-Tibetan, specifically in my research project that is also funded by the European Research Council. But we are currently working on Hmong Mien, and then we want to work on Tai Kadai in this area, in the Southeast Asian area. But I'm also trying to I have people with whom I collaborate on Dogon and Bangime. Bangime is an isolated, really interesting language. So actually in this case, I'm not the main person that makes the research, but people who actually came to me and asked me for some advice on how to code the data, how to lift the data. And so we have new collaborations that are always going on there. Now we want to also work with linguists from Buenos Aires on the language of the Gran Chaco area, which are also really interesting. We are working already on Bano Takana, also with colleagues from Peru, and on Tucano languages that are actually crossing the border to Colombia and Brazil with a colleague from Brazil. And this is all. I want to thank all the colleagues at our department. You see there's many more, so I won't even name them, and all our collaborators. Specific thanks also to Russell Gray, the director of our department. And of course, also special thanks to the organizers. This was really really nice conference so far. And thanks to you all. Muchas gracias for your attention.
Well, our last speaker, unfortunately, he couldn't come today with us. So, time for question for Matis. Okay, Matis. And let me then. Okay, first, please.
Very interesting presentation, thank you. I'm wondering if there's any sort of limit to how far back you could trace languages, and could you rebuild like a lost language using your technology?
That's a very good question. I think not, because we even do not know when language evolved, right? At the moment, people speculate between like 500,000 years ago, maybe even 1 million years ago, including Neanderthal, or maybe 200,000 years ago. and our methods currently go back up to 10,000 years, I would say. Some people say they can even extend the boundaries of our methods to 20,000 years. And the reason why the Société de Linguistique de Paris actually decided to not talk about this question again is maybe even exactly that the people already by then felt it's just too far. And there's one specific difference between linguistics and biology also that people often underestimate. We do not know if language evolved only once. It might well be that it was already, because we know certain communication systems are already present there in animals. So if you assume an early divide of populations that speak a very rudimentary language, but then we really invent, somehow they have this, the genetic drift is already there to their ability, and then it evolves. This might be possible. So people have been, most people now think language evolved only once, I have to admit that. But I would say it's not obvious, so it is.
Matis, okay, would you find, or can you tell us if there is any parallel between maybe not exactly the evolution, but how numbers and maths are generated in a society as well as language? Have you seen any relationship between maths and languages?
In fact, at our department, the people, I'm not involved in that project, they work on a database of numeral words in all expressions, all the languages of the world. Okay. And I think often I do not include 0, because we know this was invented rather lately, yeah? But it's fascinating. You have the systems that are based on 5, you have the systems based on 20, you have the systems that are irregular, like the French system and things like that, and really, really strange systems. I think that doesn't really give you any hints about the deeper physics or the deeper mathematical aspects of the numeral systems, but it shows how humans, it reflects how humans perceive the whole idea of counting. And so I find this highly interesting research. And so I think the database is already more or less ready, and there are some case studies already being done on the development of the numeral systems also. But we can actually expect some more results to pop up soon, I think. Yeah.
Okay. Very interesting. Then I would ask you, as you told us that you were going to talk, and you really did, about diversity and cultural evolution, how much different have to be the languages? For example, would you say that, for example, here in Colombia we have Paisa, Costeño, Rolo, Caleño. I guess, as far as I know, even if I don't speak German, and I really don't know Germany, I know that you have some differences between regions. Would you say that it is enough to see certain divergence between different cultures and different dialects for example, in Germany, or not enough?
Actually, you could just say like the one part would be microevolution, one part is macroevolution. Yeah, if you have the great diversity between the languages, and you know there's a certain time depth that distinguishes them. Yeah. So, for example, the Tucano language we were talking about, our estimates at the moment give us something like 2,000 years at least. Yeah. So imagine there are 30 languages spoken in the Amazonas, and they are 2,000 years old. It's amazing, right? So that they go back, and we can also trace, but we do not know the further history of them. So this is similar to the Germanic languages, maybe they also have this similar age. But if you go for Germany only, then what one can do is looking at the dialect and looking at the dialect level. And what is really important there is because that is how languages spread in the first instance. So this gives us hint on the real process of divergence. Mm-hmm. While if you have languages that have already diverged for a long time, you cannot get these processes. So you need both the macro and the micro linguistic perspective, or evolutionary perspective, similar to what scholars in biology also do. So they try to look at mutations, but they also try to look at the big results of this.
Mm-hmm. Okay. Can there be any criteria in order to say that this language is more sophisticated than this other language, for example, from its grammar?
Yeah, I think the speakers of the language usually say that their language is more sophisticated than other languages, or that it's actually something that can happen. No, I think it's difficult because complexity on one level of a language may actually sometimes be accompanied by less complexity on another level, yeah? Okay. So if you have a language that allows you to construct your words in a free order, as Russian, for example. In Russian, you don't have a word order. You can just really almost completely talk as you want. It's just really a great help because you do not need to think so often. The Germans always have a hard time because we need to put the verb at the end at some sentences and at the second position of some other sentences. This actually, it's also making it difficult for people to talk then. So you could say Russian is really good, but Russian has this incredible case system that makes it really complex. So you need to learn all the cases. Yeah, so the 6, 7, 8 cases, depending on the analysis also, including a specific case for a cup of tea, and things like that, or in the garden, a locative case, and so it costs you more time. So I would say, usually languages break down to having the same complexity as long as they can express the same ideas. And we would say that even if a language at the moment may not be able, for example, to be used for writing scientific ideas down, the speakers can incredibly fast lift their own language to the level, as we have seen by the standardization of many languages in the past, right? Mm-hmm. So even if you would say that at the moment there is a difference between two languages, because maybe one language has not really is spoken by a population that has not developed the same technology as another population, yeah, then they lack words, but they can. It takes them maybe 10 years, and they have the words as well if they want to. Mm-hmm. So lifting languages to the same level is always should be used. But this is a great miracle also. People discuss complexity a lot in linguistics at the moment, and there are also some very naive solutions. So the idea is more second language speakers, they break the complexity down. But the more speakers you have, also the more complexity you have, because you have the more variation you have. So all aspects that are difficult to. So looking at one level, yes. Looking at language as a whole, it's difficult.
Okay. Please, Joao.
Well, thanks for the presentation. Is there any expectation that linguistic lineages will match or should match recent human lineages in terms of genetics?
I think not necessarily, because languages are also really different from biological species in the sense that one person can talk many languages. So bilingualism, so you would have the same genome and different expression. I don't know how you actually, don't have a parallel here. So it's perfectly possible that you have many a multilingual society and all these cases. So it will be messy, the picture. However, of course, in many cases, it can be a good predictor to some degree. The question actually that we should ask ourselves is, so what is the least degree that it can predict, or what is the largest degree that it can predict? And in some cases, it can be amazingly well. So we have cases in Southeast Asia, I think certain cases like the distance automatically inferred distance between the languages more or less reflects the gene distance by a correlation of, I think, 0.6 or something like that, which is then quite impressive, or quite but other cases where it's completely the opposite, and we have enough examples. Also, I mean, what Cosimo, with the paper I was quickly mentioning there, is also showing that, yeah? So they are replaced genetically, but keep the language, which is completely, completely amazing also then actually as a process.
Because we can expect languages to evolve at faster rates than human populations in terms of genetics, and that there has to be what biologists call a horizontal transfer should be much more frequent in terms of cultural aspects like language. So that will diffuse any signal maybe for that matching, if there is some matching between both patterns, linguistic and genetics.
Yeah, definitely. I would say, I mean, what we hope is that we can find some examples where we can help each other in the disciplines. So I would also, so at the moment it's often that, most of in archaeogenetics, genetics, for example, many theories that are discussed now have been developed first in linguistics, because this was the first time people had data. Now we have much more interesting data, or say like data that we believe is much more safe than the speculations by some couple of linguists in the past, and sometimes they confirm what the linguists already thought. So, but if you look at the modern, also the literature and what the people use, they need linguistic labels, for example. Usually one would not just say population A and population B, although maybe that would be more properly even. But so in this sense, the problem is that, that really, so how to separate culture, language, and genes. It should be separated because it does not need to co-evolve completely or evolve completely in the same time. But and on the other hand, it's also feeling wrong to just say like we do not look at the language that the people speak when we only look at the genes from them, right? So it's a difficult question, right? Cosimo, would you agree?
We need to contextualize. No, I mean I agree with you. I mean we need to contextualize our genetic findings. Otherwise, you know, we cannot work in boxes and without seeing what the archaeological evidence is telling us, what the linguistic evidence is telling us, when it's available. I mean, if you're looking at Paleolithic time, it's not possible. You can maybe do other inferences about, you know, climatic changes and so forth. But when we have this data, it's really intriguing to compare and see if they, you know, if there is correlation or not.
Okay, then I would make my last question. And as I saw, you know several languages, so that I would ask you, are there better words to name certain objects? For example, would you call in English ant an elephant, or in Spanish, hormiguita, un elefante?
Yeah, this is also an interesting question. And the linguist would first say no, because Saussure said everything is arbitrary. Yeah, but then even people from our department were again involved in a study where they looked at sound symbolism, and that language always tend to actually use certain consonants for certain words. So the word for I, so the person, I, me, is often like accompanied by a nasal, but by a ng. And you have this actually in many Chinese dialects. You have this. It seems to be a pattern that is statistically valid. They say, they claim. So one needs to test it on more data. And there are some other interesting examples. So you have words like for fly. So it's fly here, then it's pl in other languages, or blah, blue, because you imitate the sound of the flattery. So this what the bird makes and things like that. So there are interesting patterns. But these are, of course, they are not saying that the words are better. It's just that the words are. The people, when they coined the words, this was just the easiest way to coin them, maybe in a sense.
You wouldn't say then that the sound of the word would in certain way recall the main object which is named?
I think that is still not possible. It's more in creation of new words. The cognition mechanism would maybe, for example, there's also the speculation about the E is for small and R is for large. So you have mikros in Greek, in ancient Greek, and makros. So you have a distinction there, and apparently in Japanese they have it also, these patterns. So if you would say that it doesn't mean that you will understand the words more properly, because you never know. It's similar to the uncertainty relation. Yeah, you only, if you open the box, you know whether the language really means it, if you don't know the word, right? Okay. So you cannot understand a word that you have never heard before. But it might have certain mechanisms that it's more easily, maybe it's more stable then, because it conforms more to the expectation, I don't know. But it's an interesting question also. One of the questions that need to be asked.
Okay. Okay, last opportunity. Okay, Matis, thank you very much. I then would say that, okay, thank you, Matis. Very, very good questions and answers, your questions and your answers. Then I would say that with this, we have really got to the end of this meeting. I think that talking in the name of the science faculty, we thank you very much for being here, for your talks, for your questions, and for the whole scenery of being together. So, bye.